Okay, so we're now going to move on to acquired immunity. This is the immunity that we get to things in the environment that we're exposed to a what we're going to call a causative agent. So the causative agent itself, isn't something. This is not a situation where you've gotten a vaccination. This is a situation where, like, you're exposed to somebody else with a cold. It is an active process by which the disease actually gives you immunity. Now, a cold is actually a bad example because the rhinovirus mutates so quickly that you can become immune to one type of cold virus, but it mutates and then you get exposed again and you catch another cold. Passive immunity is immunity that passes. So active is where you get immunity when you're exposed to something, all right? Passive immunity, we've talked about this briefly, is immunity that usually passes from a mother to a fetus through the placenta or through a mother's milk. Passive immunity is what babies are born with. They're born with some immune function based on what their mother has been exposed to. So it's a really fascinating field of immunology where if you see that a mother was exposed to some pathogen, and interestingly, they're seeing this with COVID-19, if a baby's mother had COVID-19 and mom started developing antibodies to COVID-19, the babies are being born with those antibodies, which is amazing. That means they're born with the protection from that disease. Now, artificial immunity is different. This is where we expose ourselves to the causative agent. So it's deliberate. So let's go back. Acquired immunity, natural immunity to things in the environment that you're exposed to. Artificial immunity, these are our vaccinations. So active is the result of a vaccination. Passive is, again, this is where you have protection being passed from one person to another. And this is a situation where you can take, let's say, again, with COVID-19, people who have had COVID-19 and whose bodies have made antibodies to protect them against COVID-19, you can harvest blood plasma from those people, purify it, and give it to people who might be getting sick. In that way, they're going to demonstrate passive immunity to COVID-19. And so in this way, their bodies aren't themselves making the antibodies. Someone else's body did that for them. And through a transfusion, they're getting protection. Next slide. All right, so how is our body actually learning to identify different things that we've come into contact with in the past that are dangerous? That's done by the development of antibodies. So antibodies are proteins that look actually kind of like this, believe it or not. Each one of these little sticks represents a chain of amino acids. And these little sticks kind of kink there. There are two light sticks here. In fact, those are called the light chains, these little ones here. These are the heavy chains, and these little bonds are disulfide bridges holding the proteins together. So antibodies actually have this kind of Y shape. The areas at the tips of the antibodies, and this is just a little extra info right here, are the areas that stick to a foreign pathogen. So let's say we have this little purple staphylococcus. This antibody would stick to two staphylococcus bacterial cells. And in this way, the body would be able to say, oh, well, wait a minute. If the antibody is sticking to these guys, we know that those guys are bad for us. Staphylococcus, staph infection. What this antibody is doing is labeling them for destruction. And so what happens is these sites on the antibodies are very specific to foreign proteins on the surface of the cells, on the surface of the staphylococcus. And yeah, I made this staph kind of small, but the staphylococcus would actually be more like maybe this size, much bigger compared to the antibody, because that's a protein, right? And so anyways, maybe I have a picture in a minute. This is what we call antigen to antibody complex formation. The thing on the surface of the staphylococcus cell is the antigen. The antibody is the antibody here. And we call that the antigen-antibody complex because whenever you have proteins coming together and sticking together, we call them complexes, all right? This is our antibody-mediated or humoral immunity. These antibodies float around, this thing in red, looking for, or not looking for because they're not alive, but they're floating around and they attach to single sites on their targets. And once that attachment happens, those targets, the cells, the bacterial cells in this case, are signaled for destruction. How does destruction happen? Well, phagocytosis may occur. So this may trigger phagocytosis right here. We can have a clump form. So like here you have kind of a little clump. A clump or an agglutination of cells can happen, which would trigger a phagocytic response by larger cells like neutrophils. And then the antibody antigen complex can neutralize toxins. So in addition to living things in our body, there are things like toxins from the environment that can get in our body that once we have immunization against them, like tetanus, let's say, those toxins can be deactivated through the antibody antigen complex formation. Here's a much nicer picture. So here's the antibody. The antibody is inactive for a period of time because, you don't want it to go crazy. Once the antibody interacts with the antigen here, so you have the appropriate antibody activation here. So here's the antigen. Here's antibody antigen forming. That starts a whole cascade with white blood cells. So this antibody in this particular case recognizes a white blood cell, and the white blood cell can cause inflammation, histamine response. Okay, so inflammation in this particular case would be histamine. So for example, when you take an antihistamine, what you're doing is blocking this response right here. Okay, and then you have another option where a cell can phagocytose the antibody with the antigen on the surface. And this little thing, this antigen here, is just going to be whatever, pretend that's a bacterial cell. Over here, we have clumping or agglutination, where you have a bunch of antibodies and antigens coming together. That also helps facilitate phagocytosis. So this process almost always goes to phagocytosis. Over here, this activation is telling cells to release histamine for an inflammatory response, which can bring in phagocytic cells. Or the other thing that can happen is T cells can come in and basically handle that thing. Okay, there's a whole separate set of proteins, which I always thought was the funnest thing in the world, called complement proteins. This is something you may not have heard about before. This is a set of proteins that we have in our blood floating around all the time, but the complement proteins are inactive. What complement is a set of, depending on the species, up to 12 proteins that basically works to lyse or burst a living bacterial cell once it's gotten into your system. It is huge for the action of antibodies, and I'll show you how that works in just a minute. So here's an example. You have a bacterial cell. Each one of these complement proteins, we have complement 1, complement 2. Don't worry, we didn't number them in this picture. They come together to form these little flower-looking structures. These little flower-looking structures are actually holes that are going to allow water and sodium to rush into the cell. And once that happens, water, so usually it's sodium that goes first, because outside the cell, you have a high sodium concentration, and inside you have low sodium. These pores, they're actually called porphyrins, cause sodium to go in because they're only permeable to sodium. So these porphyrins are gates for sodium ion entry. Nothing else can go through except for water and sodium. Once sodium goes through, though, water is going to follow it through osmosis, right? Water will diffuse down its down its concentration gradient into the hypertonic environment, and psh, the bacterium bursts. So in this way, complement proteins are directing the inflow of sodium and the secondary transport or osmosis of water to kill a bacterial cell. And this has everything to do with the complement cascade. We call it the complement cascade. It's actually a whole lot more complicated than this because there are activated antibodies involved in causing the porphyrins to form. You don't need to worry about it right now, though. We talk about that in Bio-18. Of course, phagocytosis. Phagocytes, neutrophils, monocytes, macrophages. In the liver, macrophages are called Kupffer cells. In the lungs, they're called dust cells, which I've always found funny. Underneath your skin, they're called dendritic cells. Basically, these phagocytes run into something that's foreign and eats it if it doesn't recognize it as something, as a part of the body. Now, something to bear in mind is what identifies foreign. Okay, so here's a nucleus of a cell. And for me, I have proteins on the surface of all my cells that are the Dr. Val proteins, DV. I have basically a protein, a signature protein that identifies every cell that's expressed on every cell in my body and identifies it as a part of my tissue. And that's a good thing because that means that my phagocytes look at that, say, oh, that's the Dr. Val protein. I'm a Dr. Val phagocyte. I'm not going to eat my own friends. On the other hand, if we have a bacterium, let's say an E. coli, right? E. coli has all kinds of fun little E. coli proteins on its surface. But my phagocytes are going to come up. I'm just going to draw out a phagocyte like this, right? Phagocyte's going to come up, look at Dr. Val. Not going to eat Dr. Val because Dr. Val is basically another part of its own body. E. coli, on the other hand, which is disgusting. I don't know how I got E. coli under my skin. Yuck. Got scratched by the cat or something, right? This phagocyte will not find that Dr. Val protein on the surface, and it will eat the E. coli, which is gross, right? Let's just think about, where does E. coli come from? So here's an example of phagocytosis, red blood cells attached to a macrophage. So sometimes a macrophage will consume a red blood cell if it's a red blood cell not of the body type. And so here's a macrophage engulfing the red blood cell and eventually completely digesting the, well, it will completely digest the ingested red blood cell. So it's pretty cool. Lymphocytes, these are the most numerous types of immune cells. They become B cells. B cells are in the form of memory B cells or plasma B cells, which we'll get to in a minute. But B cells start out as really primitive stem cells in the red bone marrow, and they go through several stages of development. The first stage, they're still kind of immature. In the second stage, though, at the very beginning, second stage, those immature B cells become active B cells, okay? I have a figure of this in a minute. That initiates, it's initiated by other B cells that get into contact with what we're going to call antigens. Now, one of the things you need to be aware of is that proteins on the surface of any cell is called an antigen, all right? There are other things that can be an antigen as well. It could be a toxin. It could be something on the surface of a virus. It's just a protein on the surface of something. These antigens bind to the surface via antibodies to activate a B cell. An activated B cell is going to start dividing. In other words, you have this immature B cell sitting around, and all of a sudden it gets the instructions to recognize this antigen, let's say on stomach flu virus, okay? It's going to recognize this antigen as something foreign, right? This thing right here. And it's going to tell the body, it wants to tell the body anything that has this antigen on it, kill. All right, so the B cells are the ones raising the alarm. What happens then is the B cells that are activated now to identify this thing, okay, then start dividing to make two clones, plasma cells that are going to make antibodies to identify this, to do that antibody antigen complex thing, and memory B cells. Memory B cells are stored in lymph nodes, and these are where we have our active immunity. We'll pick up again in the next video.